Hello everyone, I'm going to be talking about the new ways to access CoGe from within MaizeGDB. If we go to MaizeGDB, you'll see up in the right hand corner there's a little announcement about the new data, but let's just click through to the genome browser and show you how this works. So when the Maize genome browser loads, you'll see that there are a lot of different tracks. Right now what we want is a region that contains a gene, which we do have. So if we scroll back to the top, there will be a button called See This Region in CoGe that we're going to click. When you're referred from MaizeGDB, CoGe will greet you with this little window and a couple of links. The first one is to a walkthrough of really what we'll be doing today. The second one explains how we generated the maize sorghum analysis to make these links possible. So this is the first, this is the walkthrough. This is, a lot of the same information I'll be covering today, really in more depth. So if you have any questions, that's the place to check out. If we look at this gene that we actually came here to examine, you don't see any blue circles, and there's nothing in the annotation to suggest that there's actually a synteny there. So this is not a synthetic gene, it looks like. Let's zoom out and see if we can find one. And there's one right there. Oh, you can actually see there's another a little farther away. So we can scroll around the genome a little and spot a whole bunch of genes that are synthetically conserved in sorghum. If you click here, you can see there's a little link that describes it. These two blue circles are a good marker, but here you can see more details, including the gene names. Now what I've just done is click on the link, which takes us to GEVO, which is another part of CoGe, and it's doing a comparison of these two genes, which will be visible in a moment once it loads. Now it's loaded. Now there's several things to notice here. First of all, the yellow is the gene that we actually selected in both organisms. The green genes are other genes in the same region. I've just shift-clicked on the various BLAST-C hits so you can see how the homologous genes are distributed in the two regions. Over there you can see there's a little inversion. And one thing to notice is just how wide a region in maize it takes to occupy a smaller region in sorghum. Both of these are 400 kb, but the maize region, the whole 400 kb maize region fits into probably about 100 kb in sorghum. So let's say that we're interested in this little, well, it looks like a single inverted gene in the bottom left. What we can do is use these sliders to bring in the region we're looking at so we can really focus in on the region of interest, because right now everything's quite small. This does take a little while, but now I've got it, and we can rerun our GEVO analysis, which takes just a moment because it's a smaller region, and you can see that it's actually not an inversion. It looks like this gene may have duplicated, and there's that. We can look at that hit, and look at this hit, and yeah, so there appear to be two copies in sorghum, but only one in maize. So this may represent another copy that either wasn't sequenced or wasn't called as a gene. So why don't we turn off the CDS filters so we'll look at the entire sequence and see if the copy in maize simply wasn't called as a gene. And now you can see that, in fact, that does not appear to be the case. We've got these two little segments here, which I'm not quite sure what they are. There appears to have been a transposon that landed in maize between them. Now let's look at another couple of cool things. Here we've got a gene that is conserved in both of the homologous regions of maize. Remember, maize has a whole genome duplication that occurred after the split from sorghum, so each sorghum gene is potentially in two maize regions. And here you can see that breakdown. We've got maize and then sorghum and then a second maize on the bottom. And I think we're going to, alright, do a shift click, and so you can see the distribution of genes in maize and sorghum, and you can see sometimes genes have been pulled close together by deletions, sometimes they've been spread out by insertions. What you see here is a gene that's been retained on both contigs. We're only interested in the top one, where you can see that the part of it is actually inverted in maize, which is probably not real. What it is those yellow lines represent gaps in the contigs of the backs, and so probably that contig has simply been flipped because there's not data on the orientation. So now that we've learned a little about the new resources available in CoGe, let's try a sample research project. Imagine that we're interested in the shrunken-2 gene, which I can find using MaizeGDB. This is actually a gene involved in sweet corn production. So we click on the gene, or we found the gene location, we move over to synteny, and we see that the gene actually is synthetically conserved. So we can click on the GEVO link and move over to GEVO and do the comparison there. And with the magic of editing, we can jump right over to the gene and select the sorghum gene. And this time we're actually going to move from the sorghum gene over into annotations. And we can look at the information on the sorghum gene, which there isn't a huge amount, but the interesting thing is we can then send this over into CoGe Blast. And within CoGe Blast, you can see the sequence is already there. We've picked the various grass genomes besides maize, including Brachypodium, which isn't sequenced yet. Now, these are all the very significant hits. You can see which chromosomes they're on and how they're distributed across the chromosomes. And you can see that there are 2 copies in rice that we found, and 1 in brachy, and 2 in sorghum. And maybe there's another copy in brachy that just fell below the cutoff, the very stringent cutoff threshold I used. It's also possible that brachy has lost a copy. But now we can select each of the different hits that we've seen and send them back into GEVO again and create this panel of 5 genes, I believe. 1 from brachy and 2 each from rice and sorghum. And you can click on these links and see the information contained in there. And let's focus in on this a little more. You can see that you can see all the information you normally see about a blast hit, the percentage, the length, the e-value. If you want to, if you click on the full annotation, you can actually even see the alignment, which is going to be familiar to anyone who's run an NCPI blast or command line blast in the past. So one other thing that's important to remember is that you can use this link here at the bottom as a way of saving a specific panel to come back to later or to distribute to other people. I'll put this link up on the website so you can actually play around with it yourself if you'd like. Besides that, we're basically done with this demonstration. I hope this has sparked some interest of yours, and maybe there's some way that CoGe can be used in some of your research. And have a good day, night, or whatever it is when you're watching this.